Now let's discuss what is the procedure of IMViC test. So in the IMViC, the I stand for indole test. So let's discuss about first indole test. So first is, what are the reagents generally used in indole test? Two reagents are generally used. Number one is indole spot reagent and number two is indole Kovac reagent. So indole spot reagent include DMACA, then hydrochloric acid and deionized water. On the other hand, indole Kovac reagent include p-dimethylamino-benzaldehyde, hydrochloric acid and amyl alcohol. So these two reagents, indole spot reagent and indole Kovac reagent, are generally used in the indole test. So now the procedure of indole test. First, take a sterilized tube containing 4 ml of tryptophan broth. Then inoculate that tube aseptically by taking the growth from 18 to 24-hour culture. Then incubate the test tube at 37 degrees centigrade for 24 to 28 hours. After that, add 0.5 ml of Kovac reagent to the broth culture and observe for the presence of or absence of ring. So the result interpretation of indole test include if the result is positive, then formation of pink to red color cherry red ring in the reagent layer on the top of the medium within second of adding the reagent if the result is positive. If the result is negative, then no color change even after the addition of the appropriate reagent. Next is methyl red test. So methyl red Vox-Proskauer broth is generally used in this tube test. The composition of MRVP broth is polypeptone, glucose, dipotassium phosphate, distilled water, and the pH of the medium should be adjusted to 6.9. The procedure of methyl red test include the MRVP broth is used for both methyl red test and Vox-Proskauer test. Only the addition of reagent is different and both tests are carried out consecutively. First, inoculate 2 tubes containing MRVP broth with a pure culture of the microorganism under investigation. Then incubate at 35 degrees centigrade for up to 4 days. Add 5 drops of methyl red indicator solution to the first tube. A positive reaction is indicated if the color of the medium changes to red within a few minutes. So this is the result. The development of a stable red color on the surface of the medium indicates sufficient acid production to lower the pH to 4.4 and constitute a positive test, because other organisms may produce smaller quantities of acid from the test substrate. An intermediate orange color between yellow and red may develop. This does not indicate a positive test.